Before we start, obviously, I'd just like to acknowledge the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay our respects to their elders past and present, and the elders of any other communities who may be here today. So it's a fantastic pleasure to introduce today's speaker, John Bernardini, for his PhD oration. John joined my lab initially as an honours student a few years ago now, and it was clear from the outset that he was a very driven and talented researcher, and given that that project involved establishing not one but two new approaches to the lab, clearly he was not shy of a new challenge. And that hard work and that effort in that honours year was paid off by a well-deserved award of the Coleman Speed Medal for the top honours student of that year. So it was fantastic, obviously, over the moon when John came to me and talked to me about doing a PhD in the lab, and in particularly so because he was keen on, again, taking on a new challenge, and that was investigating a new focus for the lab, and that's the E3 ubiquitin ligase Parkin and its role in mitophagy. So throughout John's PhD, he's been fantastic to supervise, largely because it's involved actually very little work on my part. All the work he's going to talk about today has been almost entirely driven by John. And just to exemplify, we have our Friday meetings over tea and biscuits, and some of you may have seen us in the corner of the tea room talking about his work. And it became a regular occurrence that he would present a particularly nice piece of new data, and now I would seize that opportunity to jump in and, with my supervisory wisdom and obviously unique insight, to talk about what the next critical experiment might be. And to John's credit, he would indulge me for a little while, and then he would get bored of listening to the old man, and he would just slowly start to turn the page of his lab book. Just to show me that not only had he thought about this experiment, he'd actually improved it and done it on often multiple times. So, and it's great to see, again, that hard work, that diligence and that independence and drive has paid off, because we just had notification this morning, actually, of acceptance of his paper in EMBO Journal. So that's a fantastic outcome. And so I won't go on anymore, otherwise John will have to change his title. So John.
All right, thank you, Grant. That was very kind. Welcome, everyone. It's a real privilege to be talking about my work today in this seminar series. And I'm going to be talking to you today about the majority of my PhD, which has been looking at the intersection of two critical signaling pathways at the mitochondria. Now, one of these pathways you'll be very familiar with, as a lot of the work on intrinsic apoptosis has actually occurred at WEHI, as you're well aware. But the second pathway, that of mitophagy or mitochondrial autophagy, is something I think the Institute will be hearing a lot more about in the coming months and years. And so I like to begin my talks with this slide here. So these are HeLa cells, which have been stained with a red mitochondrial dye, expressing a green fluorescent mitochondrial reporter. And I do this for two main reasons. The first is to show you a nice colourful image. The remainder of the talk will be in grayscale, completely Western blots. So I like to get a bit of colour out of the way early so you can remember what this looks like. But the second most important reason is to stress that mitochondria are not the cookie-cutter organelle that are often presented in textbooks. Instead, they're part of this large dynamic network that's constantly undergoing fission and fusion. I also like to stress that mitochondria have never really been fully integrated into eukaryotic cells. And I say this because, as we know, eukaryotes in the past took up mitochondria in an endosymbiotic event. And this was because we get a tremendous advantage from having mitochondria in our cells. They generate a vast quantity of ATP for cellular processes, for example. But they can backfire. So, for example, they can produce reactive oxygen species as a byproduct of this metabolism. And also, they're semi-autonomous. So they've retained their own DNA and their own ribosomes. And what this means is when the contents of the mitochondria are exposed to the cytosol, so if these lyse, we get a really strong pro-inflammatory response. And so it's critical that the cell has a set of molecular machinery to monitor this network and to quarantine and degrade damaged mitochondria as required. So it's not all doom and gloom. We actually derive a tremendous benefit from mitochondria. Most importantly, we have this electron transport chain, which generates cellular ATP. So essentially how this pathway works, metabolites from glycolysis are fed into the mitochondria, and then they're converted to an electron, which is passed along this electron transport chain here, and this generates a proton force between the intermembrane space and the matrix. And this is then coupled to the generation of ATP. And so this is a tremendous advantage that we derive from mitochondria, but work here at the Institute and elsewhere has also shown that these mitochondria are also integrated in other cellular processes. So programmed cell death or apoptosis, we know, is finely regulated by the balance of the BCL2 family on the exterior of mitochondria. Similarly, mitochondrial dynamics play a role in particular cell types, such as secretory cells and neurons. And so the spatial arrangement of this mitochondrial network is often key to how a cell functions. And the sum total of these pathways and others determines whether a cell can proliferate or not. But each of these pathways can be dysregulated and is associated with particular pathologies. So, for example, a whole host of degenerative conditions are associated with damaged mitochondrial metabolism and the accumulation of reactive oxygen species, for example. Work here and elsewhere has also showed that defective apoptosis can result in autoimmune conditions as well as cancer. And the ablation of these mitochondrial dynamics is associated with a number of degenerative conditions as well. Parkinson's, you'll see, is associated with both defects in metabolism and apoptosis. And this will be primarily the backdrop for which I talk about mitophagy today. And so what is mitophagy? I've referred to it a few times. It's essentially the autophagosomal clearance of damaged mitochondria. And so over time, mitochondria can accumulate damage, and this is either as a result of mitochondrial DNA mutation or as the byproduct of oxidative phosphorylation, they generate reactive oxygen species. So over time, discrete regions of this expansive network become damaged. And so the cell has a set of protective molecules, PINK1, a kinase, and Parkin, an E3 ubiquitin ligase, which are the first responders to this mitochondrial damage. So how this works, essentially these two proteins act in the mitophagy pathway in concert. PINK1 is essentially a molecular sensor of damage, and it can then switch on Parkin, an E3 ubiquitin ligase, that can facilitate the selective binding and extension of the autophagosome around this damaged mitochondrion. And then through canonical pathways, this autophagosome fuses with the lysosome and its contents are degraded for recycling via hydrolases. And so what's important to note is if you look at patients with early onset Parkinson's disease, autosomal recessive Parkinson's, it's clear that these two proteins have a key role in suppressing this disease, which manifests in the brain, in neuronal cells in particular. So if we dive into a few of the statistics from Parkinson's disease, what becomes clear is when there's early onset autosomal recessive Parkinson's disease, there's a staggering preponderance for these patients to have mutations in this Parkin gene. So 50% of patients with this autosomal recessive PD present with mutations in Parkin. And further underscoring how important this mitophagy pathway is, if you look at PINK1 as well, this is mutated in a further 5% of these patients. And if you to look at the brain tissues from these patients, what you'll see is hallmarks of mitochondrial dysfunction. So there's mitochondria with reactive oxygen species accumulating, as well as a defective electron transport chain. You get selective loss of a population of dopaminergic neurons in the brain, which leads to the tremor and motor deficit associated with Parkinson's disease. And there's also a strong association with neuroinflammation. And this is something I'm going to revisit later on in the talk. And so it's clear that mitophagy is key to suppressing this pathology in Parkinson's disease. And so it's really important to understand how Parkin and PINK1 are coordinating this cytoprotective effect, particularly in the brain. So mitophagy essentially occurs in 3 steps. There's an initial molecular sensing and tagging of a damaged mitochondrion. It's then recognised by the autophagy machinery, which extends around this damaged mitochondrion once it's quarantined. And then there's the fusion of the lysosome, which leads to the degradation of the contents of the autophagosome and recycling of these biomolecules. And so for the purposes of today's talk, I'm going to be talking about this initial upstream tagging event, that's mediated exclusively by PINK1 and Parkin in neurons. And so how does this pathway work on the molecular level? So it's important to understand how this works at the steady state. So in healthy conditions, when the mitochondrial network is largely unperturbed, PINK1 is constitutively expressed by the cell, and it's then imported to the mitochondrial outer membrane via the translocase of the outer mitochondrial membrane, the TOM complex, and then it's then threaded through to the translocase of the inner mitochondrial membrane, or the TIM complex. And so when PINK1 is exposed on the inner membrane, its N-terminus is then clipped by two key proteases, one of which is PARL and the other one is MPP. And this clipping event rapidly signals for PINK1 to be degraded. So it's shuttled out of the mitochondrion and degraded via the 26S proteasome. And so under steady state, although we have constant surveillance of the network by PINK1, there's very little functional protein in cells. And we can contrast this with when a mitochondrion is damaged. So we can induce mitochondrial damage with electron transport chain uncouplers, which inhibit this aerobic respiration, and I've denoted these as AO for antomycin and oligomycin. And when we do this to mimic mitochondrial damage, what happens is PINK1 begins to accumulate on the mitochondrial outer membrane. And this is because it can't be threaded through to the inner membrane, because this critical proton gradient that electron transport chain maintains is now gone. And so PINK1 has two key substrates at the mitochondrial outer membrane, the first of which is ubiquitin, which it phosphorylates on serine 65, which leads to Parkin recruitment, and then PINK1 can secondarily phosphorylate Parkin on serine 65 in its ubiquitin-like domain. And David Commander's group and others have shown that Parkin binding to phosphoubiquitin and then this additional phosphorylation event leads to a series of structural rearrangements in Parkin, allowing it to open up and to begin conjugating ubiquitin onto its substrates. And so this essentially starts a positive feedback loop. Parkin can ubiquitinate more mitochondrial substrates, which are then in turn phosphorylated again by PINK1, which leads to more Parkin recruitment. And this is thought to be a real positive feedback loop in the signaling pathway. And so this was first visualized by Richard Yule's group a decade ago now, where they took HeLa cells, which have no endogenous Parkin, and then they expressed this fluorescently green tagged Parkin variant here. And they stained mitochondria with this red dye and damaged them over time. And what you can see over the course of an hour is that Parkin in green here redistributes from the cytosol to coat these mitochondria. You can see these nice punctate structures here, with the mitochondria encapsulated by a Parkin coat on the outer membrane. And this is important for Parkin to begin conjugating ubiquitin onto its substrates. Now, I want to just mention here that the model system for mitophagy research is using HeLa cells ectopically expressing Parkin. This is because not many cells express high levels of endogenous Parkin, with the exception of neuronal cells, and so this is the best model system we have to really unpick the molecular events of mitophagy. And so if we sort of draw this pathway together, we've got PINK1 stabilization in response to damage, recruitment and activation of Parkin, and then the accumulation of ubiquitin on substrates on the outer membrane. And this signal is then decoded by autophagy receptor proteins. These are bimodal molecules, which essentially one half of which binds ubiquitin and the other half recruits the autophagy machinery, mainly ATG8s. And in this way, we get specific formation of the autophagosome around this damaged mitochondrion. And Parkin, as the critical E3 ligase in this pathway, can conjugate ubiquitin onto many substrates. So it's important to unpick the mechanism and how Parkin does this and what the consequences are of this ubiquitination. So I've shown here the structure of Parkin bound to phosphoubiquitin solved by the Commander group earlier this year and published. And in this state, Parkin can conjugate ubiquitin onto a host of substrates. Now, ubiquitin is a small protein of about 8 or so kilodaltons, which can be conjugated onto substrate lysines via its C-terminus carboxy group up here, onto, as I mentioned, lysines. And so Parkin can begin to assemble polyubiquitin chains on substrates by essentially conjugating an adjacent ubiquitin via its C-terminus to a lysine or N-terminal methionine in ubiquitin, which I've highlighted here in this structure in red. And what that means is we can have a vast array of particular ubiquitin linkages on different proteins. And these linkages are associated with different cell fates. So K48 linked chains, for example, have been long associated with protein degradation, but emerging evidence shows that K63, K6 and K11 chains are also having a role in Parkin signaling. And so it's important to characterise the entire complement of Parkin substrates so we can get a handle on how Parkin is coordinating cell death, particularly in the context of neurodegenerative disease such as Parkinson's. So I know many of you in the audience will be very familiar with this slide, so suffice to say, intrinsic apoptosis is mediated by the permeabilization of the mitochondrial outer membrane and release of apoptogenic factors, including cytochrome C. And this is exquisitely gated by these two effector proteins, BAC, a mitochondrial resident, and its close relative BAX, which largely resides in the cytosol and is recruited to membranes, mitochondrial membranes following damage, an apoptotic stimulus, I should say. But as you would imagine, you don't want to have constitutive activation of BAC and BAX, and so the pro-survival molecules from the BCL2 family are responsible for binding to activated BAC and BAX to prevent the formation of dimers and oligomers, and also by sequestering these BH3-only proteins, which can directly activate BAC and BAX. And so it's interesting to consider this family of proteins and how Parkin interacts with them, because skewing the balance of any number of these members can sensitize a cell to death or allow it to survive. And so initial studies showed that Parkin could target the pro-survival family members, particularly by inhibiting MCL1. This doesn't really seem to gel with the fact that Parkin is cytoprotective, so when you lose Parkin, you get dopaminergic neuron loss in Parkinson's disease. So this doesn't really add up with it down-regulating a pro-survival protein. And another recent study from a group in the US, the Lavoie group, showed that Parkin could target BAX and inhibit its apoptotic function. Now, this seemingly makes a little more sense, as it agrees with Parkin being a cytoprotective molecule. But we can't quite reconcile this because we know BAX is present in the cytosol and Parkin is largely active on the mitochondrial outer membrane. And so there are a few unanswered questions here for us to interrogate, and given Grant's expertise on BAC and BAX molecular biology, this seemed like a great place to start. So to interrogate this link, I first have to introduce you to the BH3 mimetics, two compounds that have been studied extensively here at WEHI, both ABT737 and the MCL1 inhibitor, both of which activate BAX and BAC by inhibiting the pro-survival protein. So this is a really strong apoptotic stimuli. So what effect does Parkin have on BAX? So we decided to test this. We took BAC deficient HeLa cells, so these express no endogenous Parkin, and then we reconstituted them with wild-type BAX. And what we found is when we damage mitochondria with AO, these two mitochondrial poisons that depolarize the membrane, we didn't see any difference in the kinetics of cell death as read out by LDH release. And this is what you would expect. However, this changed markedly when we then ectopically express Parkin. So this is the same cell line, except now expressing a Parkin construct. Again, what we found is when we induced cell death with BH3 mimetics alone, we got nice activation of cell death that was consistent with our previous experiment. But when we turn on Parkin activity with AO, with damage to mitochondria, what we saw was a rapid arrest in this cell death. So we only got 30% of the maximum at this late time point of 6 hours with BH3 mimetics. So this really validated the data from the Lavoie group and showed that Parkin, by some mechanism, was regulating BAX activity. And so we thought Parkin's an E3 ubiquitin ligase. It makes sense to interrogate whether Parkin is able to ubiquinate BAX. And so what we did was took our HeLa cells expressing Parkin, so this model system, and then induced mitochondrial damage in the presence or absence of a proteasome inhibitor. And this would be to stabilise any K48 link chains, which might be degraded by the proteasome, for example. And what we found, surprisingly, was there was not much evidence of ubiquination of BAX at all. So there wasn't really any evidence of modification of the protein. And so we were sort of questioning whether BAX was actually directly targeted by Parkin at all. But we're kind of skewing the results here. We're looking at crude lysates. Perhaps if we pull down the ubiquinated proteins in the cell, we can enrich for modified BAX. So we gave it another crack. And so to do this, we used this assay that has been pioneered in the division by Alex and Shay in John's lab. essentially we can flow over protein lysates, some of which are ubiquitinated and some unrelated proteins, and we can then immobilize them on these cephalose beads, which bind a ubiquitin binding protein in the UBA domain. And this is via GST glutathione binding. And so what we can do is rapidly enrich any proteins that are ubiquitinated on these beads, wash them, and then determine what has been ubiquitinated in response to different stimuli. And so when we do this, we repeated the same experiment. Again, in these last two lanes, we induced Parkin activity with AO in the presence of a proteasome inhibitor. In the fourth lane, what we found was there was no modification of BAX at all, as far as we could see, even when you uncrop this blot up to higher molecular weights. This is despite the fact that we saw good modification of mitofusin 2, a canonical Parkin substrate, as well as good pull down of ubiquitin in all samples. But what was really surprising to us is when we went back and reprobed these blots, we found that BAC, the close relative of BAX, was in fact modified when we turned on Parkin signaling. And this was quite fascinating to us. It had not been reported before by any group. And so we thought this was worth interrogating a little more. So we did these subsequent experiments with a few more controls. We took our HeLa cells not expressing any endogenous Parkin or overexpressing our HA-tagged construct. And so when we don't have Parkin and turn on the signaling in the first three lanes, we don't get any modification of mitochondrial proteins, as you would expect. However, when we induced mitophagy or Parkin activity specifically, we saw mono and di modified forms of BAC. And this modification was occurring with similar kinetics to mitofusin 2 and VDAC modification, whereas an inner membrane protein, TIM44, serves as a negative control here. And so we can perform that same experiment I mentioned earlier, but this time look at BAC, whether BAC was specifically being ubiquitinated by Parkin. And this time, we coupled this assay to a deubiquitinase assay. So essentially, we can induce Parkin signaling again, and in these last two lanes, we can either enrich the population or then take these enriched ubiquitinated species and then treat them with a non-specific deubiquitinase to remove all these ubiquitin chains and thus prove that what we're seeing is ubiquitination. And indeed, this turned out to be the case. We could remove these higher weight chains off BAC as well as VDAC 1 and 2, canonical substrates of Parkin. So we're pretty confident here that we've observed a new substrate for Parkin, but we've got to keep in mind we're using quite a harsh stimulus. So inhibiting the electron transport chain with high potency might have some off-target effects. So to address this, we used a variety of different mitophagy-inducing stimuli. So we used AO that I've introduced you to before, but we also used CCCP, a proton ionophore that perturbs this hydrogen ion gradient that's critical for PINK1 import, as well as GTPP, a mitochondrially targeted HSP90 inhibitor, which causes the accumulation of misfolded proteins and induces mitophagy in a different way. And so pleasingly, when you repeat these experiments, again, without Parkin, we don't get any modification of these mitochondrial proteins. However, with all of these diverse stimuli, we saw the same pattern. Mono and diubiquitination of BAC, modification of VDAC 1 and mitofusin 2, and thus suggesting that with diverse stimulation, BAC was also a substrate of Parkin. So this is all well and good, but the question becomes, well, what's the functional consequence? We don't know that this happens endogenously, for example. So we opted for a more relevant cell line model. So we took SHSY5Y cells. So these are cells derived from neuroblastoma patients, and they have detectable levels of Parkin. And in collaboration with Iris in the lab, we generated Parkin-deficient SHSY5Y cells. So these have no endogenous or reduced endogenous Parkin, I should say. Then when we repeat our mitochondrial damaging assays with endogenous Parkin, when we expose back for longer, we see accumulation of this mono and diubiquitinated form, as you would expect. However, we didn't see this to the same extent in our Parkin-depleted cells. And this validates BAC as a substrate of Parkin in an endogenous setting, which I think was very important for us to do. And so we can now begin to assemble a model of what's happening. PINK1 is stabilised and turning on Parkin by this dual phosphorylation switch. And this then allows for BAC to be ubiquitinated. But we want to know what the functional consequence of this is. Does this have any bearing on the cell, for example? So what we did next Given that we know that BAX is mislocalized from mitochondria, we wanted to see if the same was true for BAC. So to do this, we can treat our cells with our stimulations as required, and then permeabilize them in a low concentration of this gentle detergent, digitonin. We can then spin our samples and separate them into their cytosolic fractions and mitochondrially enriched membrane fractions. And so when we do this, what we found is in a healthy setting, BAC largely resides at the mitochondria as marked by TOM20, with very little in the cytosol as marked by GAPDH. However, when we induce ubiquitination, again with AO, you see mono and diubiquitinated forms accumulating, but they're exclusively in the membrane with very little in the cytosol. So it doesn't seem likely that BAC is being mislocalized like BAX is in response to Parkin activity. And so we're kind of scratching our heads. But given we know ubiquitination is often associated with proteasomal degradation, for example, we wanted to make sure that BAC was not being degraded by Parkin. And so to do this, we extended out our time course longer. We took it out to 8 hours, as shown here. And what was interesting, although we saw rapid modification of mitofusin-2 and VDAC2, these canonical Parkin substrates, we also saw persistence of BAC when mono and diubiquitinated at mitochondria. And this is suggestive that when BAC is ubiquitinated, these ratios remain equivalent, and therefore BAC is not being degraded by the proteasome. So we're kind of scratching our heads at this point because we don't know what the consequence of this ubiquitination event is. And so we decided to go back and have a look at the sequence and structure of BAC. So Jason's kindly provided me with this model of BAC with the alpha-9 helix modelled into its transmembrane tail, I should say, modelled into the mitochondrial outer membrane. And when we look for the lysine residues, which we know are the sites of ubiquitination, we find one in the C-terminal region of BAC, which is likely inaccessible to Parkin, given that it's active here on the mitochondrial outer membrane, and a second lysine residue, which is here at position 113 in the hydrophobic groove of the protein. And we know this groove is very important from work from Grant, Peter, and Jason for the activation, dimerization, and oligomerization of BAC. And so we decided to mutate this residue and determine whether this was the sole site of ubiquitination in BAC. And it's quite rare that you get such a nice black and white result, if you'll pardon the pun, but when we reconstitute these BAC and BAX HeLa cells with either wild-type BAC or a K113R variant of BAC, what we saw when we induced Parkin signaling, when we have wild-type BAC, it behaves as our wild-type HeLa cells do, with mono and diubiquitination accumulating, but you can see that this mono and diubiquitination is completely absent when we make this very conservative point mutation at position 113. So let's take a step back and look at how BAC is activated, because this might give us a clue into why Parkin is targeting this position, this lysine in the hydrophobic groove, I should say. So by and large, BAC is inactive at the mitochondrial outer membrane, bound to the VDAC complex. And upon upregulation of BH3-only proteins, it becomes activated, as these proteins are thought to transiently bind in the hydrophobic groove of BAC, turning it on and inducing the formation of these reciprocal homodimers. So these homodimers are essentially BH3 groove swap dimers, so they're symmetrical. The BH3 domain of one BAC molecule is then sequestered in the hydrophobic groove of another. And then by an unknown mechanism, these dimers can self-associate into higher order oligomers, which punch holes in the mitochondrial outer membrane and release cytochrome C. And so you might speculate that given BAC is tagged with ubiquitin in this region, that you might be affecting this pathway of BAC activation by ubiquitination. And you can sort of flip this logic on its head and say, well, what if I induce dimerization? If I bury the hydrophobic groove with the BH3 domain in a dimer, perhaps then BAC will no longer be ubiquitinated. So we've got this hypothesis now that steric hindrance of the groove is key in whether ubiquitin or BAC homodimers can form. And so to do this, we use Blue Native Page, so we can extract protein complexes from the cell and then run them intact as shown here. So in an untreated setting, BAC largely resides as part of this inactive complex, so this 440 kilodalton VDAC complex. But when we turn on BAC, and we can use BH3 mimetics to do this, you can see this is largely collapsed down to a dimer. So in these cells, almost all of the BAC is present in this dimeric form. Again, when we switch on Parkin signaling, we don't really activate BAC at all here in the third lane. It still remains as part of this VDAC complex. But what's most interesting is if you induce the formation of dimers with this apoptotic stimulation and then turn on Parkin activity, what happens next? And what we found quite pleasingly was when you induce Parkin activity, you get this mono and di ubiquitination. But when we first induce dimerization of BAC, so we force them to form these homodimers, we could ablate this ubiquitination, suggesting that access to this lysine is key and that inducing dimerization prevents this. And this was controlled for by the fact that mitofusin 2 was modified by Parkin in this setting. So we're at a bit of a conundrum here now. We think that ubiquitination of this hydrophobic groove is slowing down BAC's apoptotic activity, but we don't really have a homogeneous population of protein to examine this in cells. And so we turned to our collaborators upstairs, and I want to say a big thank you to Peter and Jason for helping with this work. And we decided to use an approach where we can disulfide link mutants of ubiquitin as well as BAC to generate a pseudo monoubiquitinated BAC form. And this man was instrumental actually in these experiments. So we can purify ubiquitin with this G76C mutation to high purity, and we can also generate this delta N delta C BAC construct, which has a hexahistidine tail, which will be important in a few slides' time. So we can incubate these two proteins in a test tube together in the presence of an oxidant, so we use QFE, copper phenanthroline, and we can induce the formation of a disulfide bond between these two cysteine mutants. And then you can run out this heterogeneous mix of protein on a anion exchange column, and you can purify this heterodimeric adduct shown here. So this is BAC conjugated to ubiquitin as shown here, and we can get this to good purity separated away from these other disulfide link contaminants. It's important to note that this induced ubiquitination that we've generated is not truly the peptide linkage that you would see in a cell. So we know that endogenous ubiquitination involves a peptide bond between a substrate and ubiquitin, and this is a disulfide bond. However, the bond lengths are quite equivalent, and the mutation that we made in ubiquitin is up here in this G76 residue, which we know is quite flexible if you look at the protein as a whole. So now that we have our population of recombinant BAC, how can we test how functional it is? And to do this, we use the liposome system that Ahmed has pioneered upstairs. Essentially, this uses a liposome that recapitulates the mitochondrial outer membrane and encapsulates this self-quenching fluorescent dye. And some of the lipid groups in this liposome also are conjugated with a nickel ion. And this is important because it allows us to target our BAC with this hexahistidine tail onto the liposomes via this interaction. We can then flow in an apoptotic stimulus. In this case, we can use BH3 peptide and watch over time as BAC transforms from an inactive monomer to this killer oligomer. And this is able to release this fluorescent dye from the liposomes, which we can read out on the plate reader. And so when we do these experiments, we see something quite striking. So here in our positive control, this is unmodified BAC here in orange, we see rapid activation of BAC and permeabilization of the liposomes. It seems to plateau here. And what this is showing us is that we're getting a nice sigmoidal traditional activation of BAC. But when we add an equimolar concentration of BAC with ubiquitin now conjugated by this disulfide bond, you can see the kinetics of activation are much more slow. It never reaches the same maximum, and we don't have this same rapid activation. And so this seems to suggest that our pseudo-ubiquitinated form of BAC is less active on liposomes. And we can then perform subsequent rescue experiments. So we can reduce this this disulfide bond with TSEP, and you can see this largely collapses back down to its monomeric state. And when we do this, when we remove ubiquitin from this BAC, you can see the kinetics of activation largely the same, saying we've been able to rescue BAC activation and oligomerization by removing this ubiquitin molecule. And so we've got a handle now that modification of BAC with ubiquitin seems to impair this oligomerization. But we can also look at these upstream steps. And in the liposome system, we can look particularly at dimerization. So to do this, we took samples from the liposome. We repeated the same assays that I've just described. And then we ran these samples on native page and looked at the accumulation of dimers and oligomers as surrogates of activation. And what you can see is in our control, where BAC is not tagged with ubiquitin, we get rapid induction of dimers and tetramers over time. But when we add the same concentration of BAC conjugated to ubiquitin, you can see that the activation kinetics are much slower. We don't get induction of dimers at the same time frame, and tetramers are very faint and only present at the last time point. And again, we can perform these rescue experiments using TSEP to remove the ubiquitin, and we can see the kinetics of this activation largely overlay. So we get dimer and tetramer formation at the same time points. So we can go back and revisit this schematic now. We've shown on liposomes that both dimerization and oligomerization are seemingly impaired with our pseudo-ubiquitin conjugate. So what if we look at BAC activation? And this is a step that we can indeed do in cells. So this was done in collaboration with Schwai in the lab, who's pioneered this technique. Essentially, we can induce BAC activation in cells with BH3 mimetics and turn on or off Parkin selectively to see what effect this has. And so we can then permeabilize these cells in a low concentration of digitonin and flow in antibodies that are specific to BAC activation-specific epitopes, as well as a secondary antibody that is fluorescently conjugated. And then we can look at these cell populations by FACS. And so these are what the plots typically look like. You can see as a measure of BAC activation down here, we have a nice tight curve for our unactivated negative control sample. And then when we turn on BAC activation with BH3 mimetics, you can see this curve is markedly shifted to the right with almost full activation. Now, when we induce Parkin activity with antimycin and oligomycin, shown here in orange, again, as agrees with the blue native data, you don't really get any activation of BAC on its own. But what's quite striking is when you turn on Parkin and then hit cells with BH3 mimetics, what you see is an intermediate phenotype. So this curve of BAC activation sits between our negative control and our sample of full activation. And if you quantify this, it kind of looks like this. We get a strong fold change in BAC activation as measured by mean fluorescence intensity, but this is somewhat attenuated, maybe 30 or 40% by pre-treatment with AO and turning on Parkin. And this kind of makes sense, right? Because if you look at the typical blots that we get from this AO treatment, you might say that perhaps 30 or 40% of BAC in this circumstance is ubiquitinated. And so this gives us quite good correlative evidence that these modified forms of BAC are less active in cells. And so we can now revisit this and say, well, what's the sum total effect of slowing down activation, dimerization, and oligomerization? And the gold standard for this assay is to do cytochrome C release experiments from isolated mitochondria. So how this essentially works, we isolate our mitochondria from cells, and you can see here BAC is largely inactive in these monomeric forms, but we can then treat these mitochondrially enriched samples with BID, so an activatory stimulus, and watch over time as BAC assembles homodimers and oligomers that are able to release cytochrome C from the pellet fraction to the supernatant. And so when we perform these experiments in HeLa cells that don't have any endogenous Parkin, you see there's no real difference in the kinetics of cytochrome C release, and it's read out here on this graph, although there is some mild attenuation with antimycin and oligomycin alone. But what's quite striking is if you have Parkin, so in this panel here, the amount of cytochrome C release is markedly less. So you can see here, we only get faint release at our highest dose of BID when we turn on Parkin activity, as most of the cytochrome C is retained in the pellet fraction. And this is quantified here with quite a drastic reduction in a release of cytochrome C. And so we can now put forward a model of how Parkin and PINK1 are modulating apoptosis. So we know, for example, when mitochondria accumulate damage, PINK1 is stabilised on the outer membrane and it switches on Parkin activity via this two-step phosphorylation of both ubiquitin and Parkin. And what we've shown is that activation of Parkin in this way is sufficient to mono and diubiquitinate BAC and thus slow it down with activation, dimerization and oligomerization all impaired in various systems. And so if you were to consider Parkinson's disease patients, for example, who might have defective PINK1 or Parkin, these patients no longer have the capacity to downregulate BAC apoptotic activity. And so we kind of think of this pathway as a mechanism to buy the cell time to clear mitochondria and prevent cell death from occurring. And so these patients with deficiencies in Parkin, for example, have a higher sensitization to cell death and might have apoptosis, where otherwise the cell would have cleared this damaged mitochondrion. And this really agrees with some of the patient mutations that we tested in Parkin. So we took common patient mutations in Parkin, which I've modelled onto the structure here in white. And what you can see is that back ubiquination is markedly attenuated when we damage mitochondria when there are mutations in Parkin. And these mutations are associated with deficiencies in ubiquinating canonical substrates FIDAC1 and mitofusin, for example. And so in every second lane, we induce Parkin activity, and you can see when Parkin is largely inactive, it can't ubiquinate back. And so we've got a nice model here now whereby we've shown that Parkin can inhibit both back and backs by discrete mechanisms. And this is very interesting, but it kind of made us think, well, what happens if a mitochondrion is already damaged? What if apoptosis has already occurred? Is there a role for Parkin? So this was quite interesting because we know that damaged mitochondria, when they're able to persist in cells, can release these pro-inflammatory factors that I mentioned earlier in the talk. And so it might be important to have a set of molecular machines which can clear these damaged mitochondria and prevent this strong pro-inflammatory phenotype. And we're kind of thinking about this in the context of work from Stephen Tate's group, whereby he showed that at any given time with low doses of apoptotic stimuli, small regions of the mitochondrial network were damaged over time. And so you can still have your cell surviving despite the fact that a small population of mitochondria are compromised. And he showed that this could accelerate tumorigenesis in some of his models of disease. But we're kind of thinking about it in a different way. And it's not too much of a jump to think that perhaps defective mitochondria, which can accumulate in cells, perhaps as a result of PINK1 or Parkin mutations, can induce this strong inflammatory phenotype. So this was work that was done by Ben Kyle's lab and published in Cell in 2014. Essentially, what Ben's group showed was that the factor that is released from apoptotic mitochondria when they're not cleared is mitochondrial DNA, shown here in green. And this can rapidly turn on this signaling pathway via cGAS and STING to turn on pro-inflammatory cytokines such as type 1 interferons. And as a follow-up study to this, Kate eloquently showed in her science paper earlier this year that this process of mitochondrial DNA release is exquisitely dependent on back and backs. So this strong inflammatory signal that escapes into the cytosol is essentially occurring through these back pores and via herniation of the mitochondrial matrix as shown here. And so we thought this was really interesting, and we thought, well, if apoptotic mitochondria can persist in the cell and we know they can turn on strong inflammatory signals, maybe there's a role for Parkin in suppressing these damaged mitochondria and preventing this inflammation. And so what we decided to do was test whether apoptosis itself could act as a Parkin-inducing stimulus. So this slide's a bit busy. Suffice to say, we generated a number of cell lines which were either back deficient, expressing Parkin, or competent for back and backs and also expressing Parkin. And then we can turn on apoptosis again with these BH3 mimetics, these strong specific activators of back and backs. And what was quite fascinating to us is when we do this, so when we turn on apoptosis in these last 4 lanes, we saw Parkin activity, but this was reliant on 2 things. First, we had to have back and backs to induce apoptosis. And secondly, Parkin had to be present in these cells to induce the ubiquination of its key substrate, mitofusin 2. And given that we know that PINK1 has this crucial role in activating Parkin during mitophagy, it's not too much of a jump to think it might have a role in inducing Parkin's low-level activity in apoptosis. And so again, in collaboration with Iris, we generated these PINK1 deficient CRISPR cell lines. So these are both polyclonal lines with 2 independent guides. And you can see we get quite good knockout efficiency in PINK1. And so we now have PINK1 deficient lines to feed into this assay to answer the question whether this Parkin signaling during apoptosis is reliant on PINK1. And so when we do this, it's important to check these cells are still apoptotically competent. And you can see when we treat cells with BH3 mimetics, we redistribute cytochrome C, as you would expect from the membrane, marked here by TIM44, into the cytosol, which is marked by GAPDH. And This was quite a nice experiment. It was one of the last ones that went into the paper. But essentially what we show in the first 6 lanes is that we've equivalently loaded these samples, which is good. But most importantly, in these last 3 lanes, what we found is we got this low level of Parkin activity shown here in this lane. But when we delete PINK1 and then induce apoptosis, we can completely rescue this phenotype. And so we can start to assemble a model now by how Parkin's working in this context. So we know BACs are able to form these oligomeric pores, which cause damage to the mitochondria themselves. And this probably leads to localization of PINK1 near these BAC's foci. And PINK1 is then likely able to phosphorylate Parkin and ubiquitin, switching on Parkin's ubiquitin ligase activity in this specific region, localized likely to this apoptotic pore. And so when we were working through this, a paper from Richard Yule's lab just got published, and the lead author is Danielle Slater, who's actually coming to present here at WeHi next week. And her real breakthrough was she showed that PINK1 or Parkin deficient mice had this strong inflammatory phenotype mediated by the CGAS sting pathway I mentioned earlier that was dependent on mitochondrial damage. And so, unfortunately, they couldn't connect the dots between how PINK1 and Parkin deficient mice turned on this signaling, despite the fact that they unpicked CGAS and sting as the crucial mediators. But it's not too much of a hop, skip and a jump to think that what we've shown, that BAC and BAC's activation and damage of mitochondria, which Kate has visualised to release mitochondrial DNA, for example, is the link between these 2 studies. And so we now have this more consistent, more wholesome model. So Parkin seemingly is activated in response to mitophagy, but also in response to apoptosis itself. And so we kind of now think of it as a master regulator. So both PINK1 and Parkin can inhibit BAC and BACs and thus prevent them from forming these apoptotic pores, slowing down the kinetics of cell death. But this isn't their only role. It turns out that both PINK1 and Parkin are also active on apoptotic mitochondria. And you might wonder, well, what's the purpose of this? These cells are already likely going to die. But in the case of neurons, where we need these cells to persist for their entire lifetime, it might be critical for PINK1 and Parkin to switch off this inflammatory phenotype. And they may do this by inhibiting mitochondrial DNA release. And we've really shown the first evidence that PINK1 and Parkin are active in apoptotic cells. And so just to summarise, I want to highlight a few key observations that we've made throughout. Oh, everyone's sitting up, great. We've shown that Parkin is able to downregulate BACs by an indirect mechanism, but we've also shown that BAC is now a novel substrate of Parkin, and we identified this lysine at position 113, crucial substrate residue in BAC. And we showed that this occurred in the hydrophobic groove, and therefore occluding this groove with the ubiquitin molecule was able to slow down the kinetics of BAC activation, BAC dimerization, and oligomerization on liposomes and in cells. And most intriguingly now, we've got a tenuous link between apoptotic mitochondria, damaged mitochondria, and PINK1 and Parkin, and how they are able to switch off this pro-inflammatory signaling that results from CGAS sting induction following mitochondrial DNA release. And so we've really got now a bit more of a broad picture to understand how PINK1 and Parkin are regulating apoptosis and damaged cells to coordinate self-survival in neurons, particularly in the context of Parkin deficiency in disease. And so, good. I have some time. I want to say a big thank you to the man commonly referred to as the blonde Bond of WeHi. Grant alluded to our chats in the tea room, and I'm sure any of you who've seen us out there can testify that we have some pretty robust discussions. And Grant is a very thorough scientist, but he's also very creative. And it's been a real pleasure to work in your lab, Grant. I've cherished the opportunity, and I think I've learned quite a lot. And you've really given me a lot of freedom, and I think now it's starting to pay dividends, and I hope that we can really build up this project in the future. I want to also say thank you to the lab. We've been very lucky with who we've recruited, and everyone's very collegiate and friendly and helpful. And we also got Mark Lee to be productive, which was quite an achievement. I want to say a big thank you also to members of the division who've been a big help, and all our collaborators here and elsewhere who've really pushed this project forward. And I just want to highlight two key postdocs in this study, Jason and Kate. So Jason helped with all the recombinant work, and Kate recently has come in with some of our CGAS sting mitochondrial DNA angle, and these two have been exceptionally generous with their time, and they're very knowledgeable and friendly people. And I think James Murphy said it best when he said, the best collaborators are your friends, and it certainly rings true for both Kate and Jason, so thank you to them. And with that, I think it's probably time for some questions.
As John said, plenty of time for questions. Doug.
That was a lovely seminar. And it might be a stupid opening question, but I would have thought mitochondrial integrity is something that's going to be almost, and homeostasis is something that's going to be almost ubiquitously important. And yet, when you look for cell lines, you had to go and look for HL60 that had the properties. When you get Parkinson's disease, it's not catastrophic in every cell in the body.
Yep.
So why the specificity, and is there another mechanism that substitutes for Parkin more generally?
Yeah, so it's a good point. Other mitophagy pathways have been discovered. So these are associated with things like reticulocyte development and things like this. But in terms of damage-induced mitophagy, it's really PINK1 and Parkin that are key. But there's a bit of a disconnect, as you mentioned, because this really only manifests later in life. So autosomal recessive Parkinson's disease takes 20 years plus to develop, for example. And so it seems that mitochondria do have a basic machinery for maintaining their integrity. But there becomes a critical point, and I think in neurons this is key, because neurons don't have the capacity to renew, and so they need to maintain their mitochondrial network much more than other cell types. And so when this is overwhelmed, you get the neurodegeneration and the loss of dopaminergic neurons. So I think to answer your question, there are redundant mechanisms, but there's none as effective as PINK1 and Parkin. And that probably reads out in the genetic studies where if you delete either of these two genes, you get really severe early onset disease.
Following on a little bit from that question, so how is BAC expressed in the brain?
Yeah, so this is something we've looked at. In whole lysates, it's not expressed very well. There is a baseline level, but I think BAX is more highly expressed. But what we haven't really interrogated is other cells in the brain, such as glial cells or astrocytes, which might also have a role in cell death in the brain. But generally, neurons are responsive to BAX, less so BAC.
Is it possible that the dopamine-producing neurons have high levels of BAC and no one's ever actually looked?
Yeah, it's possible. I mean, I think those studies would be quite difficult to do. I think if there was a marker of dopaminergic neurons that you could isolate, by FACS or another method, that could be one way to check that, but I'm not sure.
So in your very first slide, when you showed that BAC was ubiquitous,
Yep.
you also had information, and then next to it you had a name with the proteasome inhibitor, and instead of getting more, which you probably expected, you got less.
Yeah, that's a really good point. I'll pull up that slide you're referring to. So what we saw, that's a very good point. When we inhibit the proteasome, you would expect that you stabilise K48-linked substrates. And so, for example, mitofusin 2 in this context goes up, but modified BAC goes down. And one of our early hypotheses was that this degradation was responsible for, the signalling was responsible for degrading an antagonistic dub of Parkin. So there's a dub called USP30 that resides on the membrane that antagonises this signalling, and it is also itself targeted by Parkin. So we might be getting sort of a convoluted response here by inhibiting the inhibitors of Parkin, if that makes sense.
Is Parkin upregulated in neuroblastoma as a protective mechanism at all?
Yes. So the answer to that is if you lose Parkin, you have a more severe case of neuroblastoma. So early work that we did looked at this. Our collaborators at UNSW have shown this. If you stratify populations based on loss of PINK1 or Parkin, you get sensitization. So I think it's important to have 2 functional copies of Parkin for suppressing tumour growth.
John, while you're on this slide, in a lot of the other slides that you showed, the ubiquitination that you're showing on back was mostly mono, a little bit of diubiquitination. Sometimes it was rather subtle. Once you knew that, have you gone back and satisfied yourselves that you're not seeing the same thing on backs? Like with the little higher molecular product on that band?
Yep. The answer to that is yes. Now, I should qualify my statement. When we look at backs, we see very, very, very low level ubiquitination. But we also see this, in our untreated sample. And trying to pick apart whether there's an increase or not is very tricky. And so separating this above baseline, all proteins have to get turned over at some stage. And BAX is no exception. And although we do see some faint evidence of ubiquitination, I mean, the scale compared to BAX is, it's logarithmic. But it's a good point because we sort of think of it as maybe if BAX was localised to mitochondria all the time, you might enrich this. And so you could address that, for example, by making a mitochondrially targeted BAX variant and feeding it in. But we do see very faint ubiquitination, but, throwing in lots more lysate and stabilising the proteasome doesn't seem to illuminate a role for BAX polyubiquitination.
Great talk, John. Have you tried to attach multiple ubiquitin to your back to see if you can predict the inhibitory kinetics of back activity?
Are you talking in the liposome system?
Yes.
Yeah, so we haven't done that. That proves a bit of a tricky experiment to do. So the experiment we used was this disulfide linkage system, and that's permanent and can only be reversed with a reducing agent like TSEP. As far as I know, we can't use the same cysteine chemistry to assemble polyubiquitin chains on back. There are assays you can do in vitro where you use E1s, E2s, and your E3 and then test it, but that's not something we did. And while it was suggested, it was just, we didn't get around to it in the end.
Jerry.
I didn't fully understand what your idea was about how Baxter's involved in Parkinson's. Because we think they're pretty good about such. A lot of neurosomes don't seem to have.
Yeah, it could be the case that specific populations of cells in the brain upregulate back, and that's not something you would see from a crude protein lysate, for example, and might explain why only a subset of dopaminergic neurons undergo cell death. What we're kind of leaning towards now is that back and back's already active in these cells, maybe not neurons, but supporting cells in the brain, and this leads to this pro-inflammatory effect, whereby we get mitochondrial DNA release and inflammatory signalling. So that's the kind of mechanism that we're working on at the moment, but it doesn't discount that Parkin might be active in other tissues in ubiquitinating back, such as muscle cells have been implied. And so we've been looking at the heart and things like this, but there's still a lot that we don't know.
Just one quick question. Yeah.
So is there a interferon signature in the brain of patients with Parkinson's?
Yeah, so this was addressed in the slider paper. Danielle will be more equipped to answer this than me, but I think there was IL-1 beta enrichment, IL-6, I think, and interferon.
IL-6 was the major one.
Yeah, so yeah, there seems to be this sort of inflammatory milieu in Parkinson's disease patients, and this sort of provides a bridging mechanism to why that might be, potentially. Yeah, I'm not sure. I mean, I assume that, using anti-inflammatories that could get into the brain might have an effect, but I'm not sure that's been done. Some of this work's only just been published.
So Beck.
Yeah, so the Keeler system you use is a nice experimental model, but do
Yeah.
you ever take targeted knockouts and test whether or not you get more BAX-driven apoptosis or BAX-driven cytokine?
No, I don't think we did that. That could be a good experiment to do. I mean, we used the neuroblastoma model, but again, there are some issues with that model. I think the next step is to go into mice. So Grant has generated a Parkin-deficient mouse now, and so examining some of these apoptotic effects in the MDMs, for example, or neuronal cells. So Andre in the division has expertise in this sort of cell culture work. That's the next step. But I would agree with you, using these HeLa cell systems is just an indication. It's not a complete guide.
Okay, with that, just remains to thank John for a fantastic talk.